All right, let's look at another one of these pathways, another one of these signal responses. As we just saw, you know, uh, G protein coupled to receptor triggering a calcium response or a calcium release response. So we're going to be looking at how to get certain genes expressed. We're going to be looking again at that the ERK that we talked about, that ERK protein pathway that we mentioned earlier, where receptor gets activated, starts to cascade through RAS and a few others, which activates MEK, which activates ERK. ERK traffics to the nucleus, triggers gene expression. Well, what we see happens is that these series of proteins that we talked, the MEK, the ERK, they're part of what's referred to as the MAP kinase pathway. MAP, MAP, mitogen activated protein kinases. You'll see it, MAP kinase, MAPK pathway, depending on the book, depending on the paper. So what happens is through that series of proteins that get activated, activate others, and so on and so forth, ultimately activating ERK. ERK traffics to the nucleus, where it's going to interact with, as I said, different proteins there. MNK1, which in turn, gets activated, which activates ELF4E, and so on and so forth. So what we see happens is that the ERK, once it's in the nucleus, is going to interact with this whole complex of proteins. In cell biology, when we say a complex of proteins, it means it's an assemblage of proteins that have to come together to work together towards a common goal, a final goal. Each protein in the complex has a different role. You remove one of the proteins, complex does not function. Okay, individual proteins are, mature proteins have their own tertiary quaternary structure, and it's just that to get the ultimate outcome, they have to all be together in a specific order, in a specific, laid out in a specific way to get things to work. So what you see is, as you notice by the figure, ERK, which is phosphorylated, enters in the nucleus. It will then activate MNK1 by phosphorylating it. Phosphorylated MNK1 activates ELF4E, which then interacts with 4G, 4A, EIB, which then will change the shape of certain mRNAs. Turns out mRNAs, remember the single strand of RNA that takes the copy of the gene from the nucleus out? Well, the single strand RNA is very unstable. So to stabilize it, RNAs will form weird structures. Weird structures, as you see right here. It's called a hairpin. Looks like hairpins, bobby pins, for those of us guys who have ever seen those or don't know what they're talking about. These were recognized back 4 or 5 decades ago, back when hairpins for women to keep their hair up in certain structures and certain shapes was all the vogue. So what we see happens is the complex of proteins, it comes together, and it's sitting there associated with an mRNA, but it can't do anything because the hairpin right here is blocking it. ERK comes in, activates, which allows the complex to flatten out the hairpin, and now everything can progress, move through. What are the responses going to be? Well, like I said, there's too many to count. Just talked about the last 2 slides. You get protein expression, gene expression. You can also get increases in cellular metabolism. Adrenaline. Adrenaline activates certain, what is it? Beta adrenergic. Sorry, I haven't had enough caffeine, can't talk. Receptors. These receptors will activate adenylyl cyclase, which means the cyclic AMP levels in the cell are going to shoot up. High levels of cyclic AMP activates PKA, protein kinase A, which then goes and activates other enzymes, which will then go and start releasing glucose stores. Stores within the cell, or it'll go and start cutting up glycogen, releasing the individual glucose molecules that are stored as glycogen in your liver, in your muscles. So increase in adrenaline, fight or flight, gotta have the energy, so you release the glucose, start the making of, ATP from cellular respiration. Also going to have cell growth. Different pathways, different receptors are going to trigger the cells to begin increasing in number, going through mitosis, start making proteins, start copying DNA, getting ready for mitosis. Another great example is what is referred to as apoptosis, or you'll hear people refer to it as apoptosis. Apoptosis is cell death. Within every cell in your body is the instructions on how to commit suicide. All the enzymes, all the proteins are there, and they're just waiting, but they're kept in check because the cell is healthy. Any damage to the cell that can't be repaired, those cells stop protecting the cell against cell death. Cell will just commit suicide. Better for the cell to die than the tissue. Better for the tissue to die than the organ. Better the organ than the organ or the organism. We also see apoptosis comes into play during fetal development. We start off as a blob of cells that will then form a hollow sphere. Part of the sphere will push in so that you now have layers of cells that are going to become different tissues. When you look at your fingers, your toes, turns out during fetal development, instead of a hand with fingers, you had a flipper. That skin tissue between your fingers, between your toes, was actually all the way up here. You've seen pictures of that have webbed fingers, webbed toes. Turns out that tissue's all the way out through here, and the bones are pushing through. At a certain point during fetal development, all those cells there that were in that tissue are told it's time to die. They literally call this a death signal. It's a certain protein that binds or a certain ligand that binds the death receptors and tells all those cells to die off, and then that tissue just goes away. You now have individual fingers.